Hello everybody, myself Dr. G. Mangai Kasturi, Assistant Professor of Botany, Sri Meenakshi Government Arts College for Women, Madurai. Welcome you all to this topic on optimization of plant biotechnological tools in improving plant sustainability. Here I'll be dealing with the various tools that are involved in the production of transgenic plants. My topic I have divided into 5 different sessions. 1. I'll be telling about the introduction. 2. I'll be discussing about the history. 3. I'll be discussing about the applications. 4. I'll tell about the advantages of transgenic plants. 5. I'll tell about the various methods of gene transfer. So, as we all know that several years farmers have been altering the genetic makeup of the crops that they grow. Several biotechnological approaches such as hybridization, mutagenesis and polyploidy are available for the crop improvement, but the most potent approach is the genetic engineering or gene modification. So, the plants obtained through genetic engineering contain a gene or genes usually from an unrelated organism. Such genes are called transgene, and the plants containing transgenes are known as transgenic plants or genetically modified plants, the GMP. The process of introduction of desired gene of interest or the exogenous DNA into the genome to create and to maintain a heritable character is called transgenesis, right? So, transgenic plants are those which contain the transgene, which is otherwise called genetically modified plants. Next, coming to the history. In 1982, the first genetically modified antibiotic resistant tobacco plant was produced. In 1987, insect resistant tobacco plants were produced by introducing the insecticidal protein from Bacillus thuringiensis. In 1994, flavor saver tomato were approved. In 1994, herbicide resistant tobacco plant was marketed. Again, in 1995, BT potato was approved, which was the country's first pesticide producing crop. In 1995, canola oil composition was modified. The BT cotton as well as the virus resistant squash were approved. In 2000, vitamin A enriched golden rice was the first food with the increased nutrient value. Next, coming to the applications. The transgenic plants have various applications. So, you can produce transgenic plants resistance to biotic stress. Biotic stress means insect resistant plant, virus resistance plant, fungal and bacterial resistance can be introduced. At the same time, resistance to abiotic stress, the herbicide resistant plant. Next, they also have other applications, that is the crop yielding quality, right? That is extended shelf life, improved nutrition, improved coloration, and they also can be used for the production of low cost pharmaceuticals, that is the edible vaccine and the essential proteins. So next, coming to the advantages of transgenic plants, they have tremendous advantages. There are advantages for the producers who produce new variety. At the same time, highly efficient plants can be obtained. It is also advantageous for farmers, that is the damage due to pests is reduced and the destruction of the crops can also be prevented, and the production and profit of the transgenic plants which they produce will be high. Next, for the industry, how it is advantageous is, for example, low lignin content wood can be produced, thereby it leads to the less pollutant from the paper industry, and also human protein production. Next is, they have also got other advantages, that is, for the consumers it is more nutritious, at the same time extended shelf life, can be easily stored, right? So these are the advantages. So till now I was telling you about the terminology transgene, transgenesis, transgenic plants, the history with few examples, then the application, then the advantage. Next we'll go into the methods of gene transfer, or the tools that are used in transferring of the gene of interest into the plant. It is categorized into three major division. One is biological method, another one is physical method, another one is chemical method. So first let me deal about the biological method. So how this agrobacterium tumefaction can be used in the gene transfer technology. So this agrobacterium tumefaction, as we all know that it is the nature's smallest genetic engineer, and I'll tell how this is, why and how this can be used as a genetic engineer, or why this is called the nature's smallest genetic engineer. So to tell a few ideas about agrobacterium, it is a soil bacterium, gram-negative bacterium, rod-shaped, motile, and it is found in the rhizosphere. So it is a pathogen, right? So it will cause the crown gall disease, and it affects 95% of the dicot plants. So here you can see the formation of the crown gall. So this diagram, you can see that the normal plant, it has got the crown gall disease. How this has acquired this disease, right? So the agrobacterium which is present in the rhizosphere. So the agrobacterium which is present in the rhizosphere, right? So when an incision is formed and the cut or wound is happening in the plant, this agrobacterium enters through the wound, and this agrobacterium which has got the chromosomal DNA and the TA plasmid. This TA plasmid plays a major role in causing the crown gall disease. This TA plasmid has a segment called the tDNA. This tDNA gets excised, gets inserted into the chromosomal DNA of the host. So this cell becomes the transformed plant cell. So because of the integration of the TA plasmid, that is integration of the tDNA from the TA plasmid, this plant gets the disease, the crown gall disease. So this crown gall formation is because of the presence of plasmid, agrobacterium, and it is known as TA plasmid, or the tumor inducing plasmid. So in this diagram you can see that, so the agrobacterium, 0 day its infection, 3 days the slight division of cells, you can see 18th day you can see some more gall formation, and in the 24th day it becomes a tumor, right? So next, the agrobacterium. So about the agrobacterium, it contains bacterial chromosome and the TA plasmid, right? So why this is called the genetic engineer? Already I told you, I'll tell you why this is called the genetic engineer. It transfers its genetic material into the plant, right? So it is called the natural genetic engineer. Here's where the scientists thought that when it can excise its tDNA into a host and cause a crown gall disease, why not we interfere in this portion of the tDNA and insert our gene of interest and make this to excise our gene of interest into the plant, so that it will produce a transgenic plant. So let's see what are the features of this agrobacterium. It's very interesting to know about this. What are the features, what are the functions of these features, and how it produces crown gall, and now how they modified this region and make it to work as a tool for transferring the foreign gene or our gene of interest, right? So the TA plasmid, right? This TA plasmid you can see in the diagram, right? It's a large conjugative or the megaplasmid of 200 kilobase pairs. It has got various features like tDNA region, Vir region, origin of replication, conjugative gene, and genes for opine catabolism. Right. So let me deal about each features. Right. Each feature. One, the tDNA. The Ti plasmid contains the tDNA and it is 24 kilobase pair. It contains the left border and the right border. You can see the tDNA region in the diagram. It contains the left border and the right border. Right. The tDNA contains the auxin, cytokinin and the opine. Right. And this are not expressive in the bacteria, but it is expressive in the plant. So next feature is the Virgen. These Virgens are responsible for the excision of the tDNA into the host plant and it gets integrated, right? The Virgens are present outside the tDNA region. The previous slide you have seen the diagram that it has got, the tDNA portion has got only the left border, right border, auxin, cytokinin, opine. So the Vir region is present outside the tDNA region. It is present in the Ti plasmid only and it is 40 kb. It contains 35 genes arranged in 8 operon. So this Virgen does a marvelous, fantabulous work so that in coordination of all the Virgens, this set of genes work in integration and see to that the tDNA gets excised and gets integrated into the host genome of the plant. Thereby it causes the crown gall. So I'll explain in the latest slides that how this Virgen helps in the excision of the tDNA, right? So the Virgens are 35 genes arranged in 8 operons, right? So the Virgens are silent till they are induced by certain plant factors or the chemoattractants, right? Next is about the Vir A. The Vir A senses the chemicals secreted by the plant cells. So what happens? The Vir A senses the chemicals secreted by the plant cells. The Vir A gets autophosphorylated. So the function doesn't stop only with the autophosphorylation of Vir A. The Vir A gets autophosphorylated, activates the Virgen. So already I said you that there are a set of 35 genes arranged in 8 operons, which does a marvelous work, right? So each in integration with each other, in coordination with each other. So Vir A, it is senses the chemical secreted by the plant cells, right? And this gets autophosphorylated and it activates the Virgen, right? So activation of Virgen induces the expression of all the Virgens, right? So activation of Virgen induces the expression of all the Virgens. Now, what is the role of Vir B? So the Vir B produces energy required for gene transfer. I discussed about the tDNA, which is one of the feature. Next I am discussing about the Vir, which is outside the tDNA. It includes the Ti plasmid, includes the Virgen and the tDNA. Right. Now I am telling you I am discussing about the features of the Virgens. Now coming to Vir B gene, it produces energy required for the gene transfer. Vir C enhances gene transfer into the host cell. Vir D, there are protein variants D1 and D2. This D1 and D2 exhibits enzymatic activity, that is topoisomerase and endonuclease activity respectively. So next is the Vir E. So what does this Vir E do? Again, these are also protein variants E1, E2. They help in proper folding of the DNA. Ultimately, their aim is to excise the tDNA from the plasmid and make it reach to the host so that the host gets the crown gall disease. So As soon as it senses the chemical, the Virge, it starts activating the Virgi. The Virgi activates all the set of genes so that ultimately together what they'll do, they will excise the tDNA, make it integrated into the host genome, right? Virh arrests the cell division. Virh and Vir-D2, what they'll do is transfer the single stranded DNA into the host. Now, all these I explained about the features. Now I'll tell you the molecular mechanism of the tDNA's transfer. Here I'll be telling about the transfer and integration of the tDNA into the plant genome. Step 1. What is step 1? Already I have told you. It's just a flowchart which I want to explain you and make you understand and remember it forever, right? Step 1 is signal recognition by the agrobacterium. So the wounded plant cell, I've told you already, it releases the chemical attractant, the chemoattractant, which is the acetylserinone or the hydroxyacetylserinone. So we know that this chemical is sensed by the virgins, right? The Virge. So Virge, Virgi helps in sensing the signal. Thereby what happens? It doesn't stop there. Once it senses the signal, the bacteria will move towards the wounded site, right? So it will not be just near to the plant. It will be elsewhere in the rhizosphere, somewhere around it. Once it's the Vir A and Vir G senses the signal, it makes the bacteria move towards the wound, right? So now step 2. So you can see in the diagram, the step 2, the agrobacterium attaches with the plant cell. So in order to attach the bacterium to the plant cell, one, it is the initial attachment via polysaccharide. 2, because of the CHV genes, that is CHVA and CHVB genes, which are involved in the attachment of the agrobacterium to the plant cell. So this is how it gets attached to the plant cell. First is your signal recognition by the agrobacterium. Second is the attachment of agrobacterium to the plant cell. Third is the Virgen induction. Virgen induction is the Vir A, we know already, Vir A sensing, Vir A autophosphorated, triggers the Virgi, Virgi triggers all the virgins and thereby induces the expression of all the virgins, right? Next, so T strand. The virgins, Vir B, C, D, E, F, G, H, they are present in the cytosol. So we saw the protein variants of D, protein variants of E, the function of Vir H, right? And these, the functions of all these, right? So Vir D1 binds to right border and relaxes the supercoiling. Vir D2 forms the nick at the right border and binds to the 5' end of the single strand DNA, and this helps in the excision. Next is transfer of tDNA from the bacterial cell to the plant cell. So this is mediated by Vir B and D4. These two together forms the channel, the conjugative pilus or the T pilus. So the DNA is carried by Vir D2 and the integration of the DNA is helped by Vir E2, right? And this E2 protects the DNA from degradation by the host enzyme. So Vir D2 navigates the DNA into the nucleus. Inside the nucleus, what happens is the single stranded tDNA gets converted into double stranded tDNA and gets integrated into the plant genome. This process of integration is called recombination because it does not involve or depend on sequence similarity, right? So this is called the illegitimate integration. So forms the membrane channel, right? includes Vir-B protein VB2, B5, B7, which forms the T pilus. It is the major, B2 is a major pilin protein, and other Virgens also pass the T pilus in the plant cytoplasm to form the mature T complex. So this is the diagram. So what I have told you theoretically, it is in the pictorial form. That is, one is the plant derived signal are recognized. Second is Virgen activation. Virgen activates all Vir-B complex. Then the substrates are recognized by Vir-B complex, right? A T complex assembles, and next is the tDNA getting integrated in the host chromosome. So the plant cell, and this is the agrobacterium cell. How the transfer and integration happens into the host plant genome. Right. Next. So these are the features, and these are the functions of these features wherein the tDNA gets excised, gets integrated into the plant genome. Hope you all understood till here the mechanism of transfer of tDNA from the TA plasmid because of the Virgen into the host plant. Right. Now, how this agrobacterium can be used for plant transformation. This is just a rough sketch of understanding. I'll tell you in detail how to go the production of the recombinant plasmid, right? So the agrobacterium tumefaciens, the TA plasmid, it has got the tDNA region. We are going to prevent the oncogenicity, and we are going to insert our gene of interest into the plasmid, right? And we are going to produce the recombinant plasmid. So by the production of recombinant plasmid, we are going to introduce into the plant cell in the culture. Now the tDNA is having the new gene within the plant chromosome, and it regenerates into a new plant with a new trait, right? So now how this agrobacterium can be used as a vector. So how this can be achieved, or how this agrobacterium tumefaciens can be used as a tool for transferring the gene of interest. So if you stop the tumor forming quality of the agrobacterium, we can achieve the transfer, right? So this tumor formation is due to the tDNA. So we should disarm the tDNA, prevent the oncogenicity, that is the gall formation, and we should catabolize the catabolizing genes can be deleted, that is opine synthesizing catabolizing genes can be deleted. Nitrogen and carbon source can be supplied in the medium. So how this can be done? This can be done by the formation of binary plant transformation vector, right? So how this can be done is, so transformation procedure is recombinant small replicon is right? A small replicon is made, which is transferred via bacterial conjugation or direct transfer to the agrobacterium. Already the agrobacterium should harbor a helper TA plasmid. So what I want to tell here is, what is that small replicon? That is, that small replicon, it has got the foreign DNA in the place of DNA, in the place of tDNA, the left and the right tDNA borders, selection and maintenance in both E. coli and agrobacterium, a selectable marker for the plant, and the plasmid is disarmed. That is, that already I told you, tumor inducing genes are removed, right? Now after that, so this is the small replicon. So this small replicon is transferred to agrobacterium. Already I have told you binary vector, right? One is the small replicon, right, which is having a wide host range, and this called the mini TA plasmid, and another one is the helper TA plasmid. So now what they do is, the recombinant small replicon is transferred into agrobacterium already, which is having the helper TA plasmid. Helper TA plasmid is which helps in the excision of the desired gene of interest. So the plant cells are co-cultivated with the agrobacterium to allow the transfer of the DNA, recombinant tDNA into the plant genome. Transformed plant cells are selected under appropriate condition, right? So this diagram represents the wide host range replicon, and this is the helper plasmid. So that replicon is transferred into the agrobacterium, which is already having the helper TA plasmid. Now this is co-cultivated along, right? So after it gets co-cultivated, then it is subjected to screening, right? So that is about the agrobacterium tumefaction. The features of agrobacterium tumefacients. So this agrobacterium tumefacient is one of the biological method of gene transfer. I've told you what is agrobacterium, what are the features of agrobacterium, and what are the functions of those features, and how it causes crown gall, and how it is manipulated to make it a useful tool. That is why it is used as a tool. How it can be the tumor inducing oncogenicity is prevented, stopped, or deleted, and we can insert our gene of interest and introduce into a plant so that we get a transgenic plant, right? This is about the agrobacterium tumefacients. Next is how plant virus, or what are the plant viruses which can be used as a vector. These plant viruses are considered as a efficient gene transfer because it infects intact plants, right? And they amplify and get transfer the genes. So those two viruses, one is virus, which is circular double stranded DNA, and 15 viruses are there, among which cauliflower mosaic virus is a very important virus for the gene transfer, right? And next is the Gemini virus. This Gemini virus contain one or more two circular single stranded DNA, and this can also be used as a plant virus vector. So next is the physical method. The first one is electroporation. So electroporation is, it involves the use of high field strength, the electric pulse, and the pores are created in the cell membrane, making it permeable for the uptake of the DNA. How this is used to deliver the DNA, right? So we'll see that it's used for delivering of the DNA into the intact plant cells and the protoplasm. That is, this high electric impulse is given, right? So this is the electroporation, wherein which we can transfer the DNA into intact plant cells and the protoplasm. The plant material is incubated in the buffer solution containing desired foreign DNA or the target DNA and subjected to high voltage electric impulse, right? So pores are formed in the plasma membrane through which the our gene of interest or the foreign DNA or the target DNA enters and gets integrated in the host cell genome. So this is electroporation, which involves the use of high field strength electric impulse, right? So pores are formed. Now because of the pores, it can easily uptake your gene of interest, which is present in the buffer solution, right? So next is the particle bombardment method. The particle bombardment method, it is a very effective way of transferring the gene. The particle bombardment method is otherwise called microprojectile bombardment or biolistic or gene gun, right? So here the foreign DNA is coated with tungsten or gold particles. That is, the tungsten or the gold particles are coated with the DNA and which are carried by the macrocarriers into the stopping screen, right? So as soon as they reach the stopping screen, these macro carriers which carry the DNA, right? They hit the stopping screen. The DNA coated with the gold particles are propelled through the screen into the target cell which is present at the base. So, right, this is how particle bombardment method is done, right? So, it is otherwise called microprojectile bombardment or the gene gun. So, it is a direct physical method which involves the mechanical insertion of the next is the microinjection. Microinjection is a direct physical method, involves mechanical insertion of the DNA. Transfer of the DNA, transfer of the gene is done through micropipette into the cytoplasm or the nucleus. So, this is the microinjection, which is the physical method of gene transfer. So, here the mechanical insertion of the DNA through the micropipette. Right. So, next one is the liposome mediated transformation. You can see in the diagram the DNA, the liposome. So, what happens here is these liposomes are circular lipid molecule with an aqueous interior that can carry nucleic acids. It adheres to the cell membrane and fuse with them to transfer the DNA fragments. Thus, DNA enters the cell and then to the nucleus. Right? So, I'll just tell you again. Liposomes are circular lipid molecules with an aqueous interior, so they can carry the nucleic acid. So what happens is these liposomes adheres to the cell membrane and fuse with them to transfer the DNA fragments. Thus, so once they fuse, what happens is the DNA enters the cell and then to the nucleus. So adhesion and its fusion at the site of attachment, they release the DNA into the nucleus, right? So this is about the physical method. Next is the chemical method. The first one is polyethylene glycol mediated transformation. Another one is DEAE, dextron mediated transfer, right? So in this PEG, polyethylene glycol, so here it is polyethylene glycol mediated transformation. This PEG destabilizes the plasma membrane, makes it permeable, and hence helps in the uptake of the DNA. Right? Next is the DEAE, dextron mediated transfer. So this DEAE is water soluble, right? And when added to the transfection solution containing the DNA, by endocytosis, the DNA uptake takes place, right? So, what are the genetic tools we have studied is, I will revise just again. One is biological method, which includes agrobacterium tumefaciens. Next one is plant virus vectors. Second is the chemical method. The chemical method we'll be discussing about the electroporation, right? Then the ballistic gun, the microinjection, and the liposomes. Then the chemical method, the PEG and the DEAE. Hope you all understood, right? So these are the various methods by which the gene can be transferred into the host plant, that is our desired gene of interest can be transferred into the host plant to produce a transgenic plant, or the novel character can be introduced for the production of the novel plant, right? Hope you all are benefited by this. And what I want to conclude is the transformed plants. So why do you go for this? They help to meet the human demands. What are human demands? Food, nutritive value food, high nutritive value food, right? The shelf life, the production of edible vaccine. So you eat a fruit, you get the vaccine. Then to meet the needs, the next need is energy, medicine, shelter, clothing, and the foremost is a cleaner environment. Whatever we eat or the energy we get or the medicine, the first and foremost is a clean environment. The clean environment is a place where we should live. The clean air, the clean water, the clean food, and the happy life, right? So next is, this is used for this gene technology can be used in making the plant to accumulate the toxic metals. So we all know that there are various pollutants which are polluting the soil and the water. It's our own, to meet our own needs, right? So what we do is we go for chemical fertilizer. So what, the more chemicals we add to that, it gets accumulated. So what we can do is we can go for a production of a transgenic plant, wherein which the plant is capable of accumulating, that is the hyper accumulating toxic metals from the soil and water. The ultimate is we get a cleaner environment. So the prime is the cleaner environment, right? Next, so this is significantly advantageous over traditional plant breeding methods, right, for the production of improved crops. Hope I have conveyed all the material, the concept of the production of the transgenic plants. Hope any of you will become a scientist and use any one of these tools to produce a more wonderful, useful transgenic plant which will meet the human demands. Thank you so much.